Good evening. Can everyone hear me?
Oh,
Oh, a little too much? Yeah, can I speak? Yeah, am I good? Oh, okay, I'm ready. All right, how's everyone doing? Having an excited time? Can I get a clap? Excellent. Okay, so howdy, my name is Walter Patterson. I'm the Chief Scientific Officer at MiniCircle. We're currently running a phase 1 trial down in the Prospera, Honduras. I'm not really going to talk about that. Rather, what I want to do is I want to inspire everyone in this room to make their own viruses, to make their own plasmids, to go out and actually do something for themselves and for the community and enrich the lives of those around them using biotechnology. So I got a lot to go through. Some of it might be a little bit boring, but essentially what I'm going to go through in this talk is showing you all the different tools. You know, if you're thinking about building a house, you use saws and hammers. Each one of these things does a different task, and it does it in a particular way. So I'm going to give you some genetic tools in your tool bag and be familiar with them. And at the end, I'll show you some resources, both free and paid for, that you can use to actually get a hold of these constructs, to actually code these things up, to play around with these kinds of things. So that way, after this little talk, y'all can go home and actually work on it, start to figure, and practice and learn. So who's ready for some genetic engineering?
yeah. Good.
Alrighty. So I know, we're all big on dogma here. Biology's no different. So the big dogma in biology is that DNA makes RNA, makes proteins. That's the whole streamline of event. And so any point of bioengineering, we can actually go into one of these 3 sections and have a useful product. Whether that, in the case of DNA, have any of y'all had the Beyond Beef burger?
Not
Tasty, right? It's a nice, tasty burger, but it's completely plant-based. They took the heme protein and put it into soybeans via agrobacterium. You can make adenosine viruses, which most gene therapies are. Anyone know about the COVID vaccines being RNA? Yeah, we all lived through that one. And even like down at the protein level, you have insulin, hormonal stuff, like all these things that you can have readily access and available to you. Many diabetics would die without it. So there's a bunch of different types. Essentially, I've gone through that. You can either have things that are in vitro use, where you're isolating proteins. You can have in vivo expression, for example, our gene therapy expressing follistatin. You can have a variety of different methods. You can have them additive or subtractive are the big 2 ones. For example, with CRISPR, you can go in, you can cut out a defective bit of DNA, or you can stop the excretion of a particular protein in a problem area. They've been looking at things like sickle cell anemia, these kinds of things, or you can actually add beneficial genes as well through an additive process. So I'm going to go through one by one of these different methodologies. So by the end of this, we'll see everything that doesn't breathe, such as bacteria, to mammalian cells, to plant cells. So we're going to have a good idea of what we want to do. So let's get into it. So just as a little bit of overview of what we'll go through. We'll go through the basic bacterial plasmid, agrobacterium plasmids for plants. We'll go through mammalian plasmid constructs. There's going to be a lot of glowing stuff in this slideshow. We're going to go through RNA, circular RNA, AAV, mini-circles, and mini-intronic plasmids, just to name a few. It's going to be a while, folks. So, plasmids. They're DNA-based, they're circular, and they're essentially the workhorse of like most gene therapies. If you look at AAVs, so viruses, they start their lives out as plasmids most of the time. If you look at mRNA vaccines and all that, you all essentially create that RNA sequence from DNA at the production level. So this is essentially the wooden handle of gene therapies. Everything at some point uses a plasmid when making the constructs for its various needs. So this is just an example of a plasmid, and we're going to go through each one of these parts and what they mean. So later down the line, when you are actually constructing your own constructs, you can easily recognize the little bits and what they do, essentially. So most of these we put into bacteria. It's real easy. You just heat them up, add a little bit of salt, heat them up again, shrink them again, add the DNA, so on and so forth, in what's called heat shock. So this is typically done in E. coli just because it's easy to grow. But the problem with E. coli is we need a way to select all of them, which has the plasmid, which doesn't have the plasmid. So we do that by a selection marker. In this case, the selection marker is an antibiotic resistance gene. So that way we can just add antibiotics. The only thing that's going to be in that solution is going to be the resistant E. coli. It's not going to be anything else, because otherwise it would die. Self-selecting. The next thing, origin of replication. This is where essentially this gets made by the DNA polymerases, actually the mechanisms inside the bacteria, in order to make more of the plasmid. That way it just doesn't disappear after the first first cell division or something like that.
Yes.
Now this is going to be something we're going to see over and over and over again with all these different constructs. Promoters, operators, these are essentially things that tell the DNA polymerase and everything, hey, I need you to code this thing in front of me. In this case, it's going to be the transgene of interest. Now, these things can be controlled. For example, you can have them on a sugar, such as the lac operon. You can have them on a tetracycline response element, or they can just be a strong promoter, which we'll see later on, which is just going to make as much protein as it ever wants. Trust me, the glowy things are coming up.
Sure.
And of course, the transgene. In this case, the hello world of gene therapy, the thing that everyone does, GFP. So green fluorescent protein, it glows green. It does what it says on the tin.
Cool.
So, like I said, plasmid use cases, you can get E. coli to make yourself insulin. You can make them, I don't know, anyone into bodybuilding, HGH, this kind of hormonal stuff. You can also use it to manufacture other components, such as CRISPR. You can have them make AAV viruses for cells. You can have them make mammalian plasmids. Essentially, anything you can think of, it almost all starts at the plasmid level. So this is I thought I had some E. coli, never mind. So this is a agrobacterium plasmid. It's the same kind of thing we just saw. This is just pulled from the website. Now, there's a couple different points on this that actually we'll go into, but basically this is also expressing GFP, and if I skip a few more slides, how many of y'all seen grass or plants, that kind of thing?
Yep. What the give me some?
Okay, a few people in here? Well, they typically don't do this. As you can see, what's different about this?
Well,
Yeah, it's glowing. You don't see that every day. Just as an example of how you can actually get these things doing. So, this in case is from the Glowing Plant Project. These are tobacco plants. So, I know this is going to sound really basically what you have on that last plasmid I just showed you is there's this whole complicated signaling process where if you put that plasmid inside the agrobacterium in the same manner that you would do in E. coli, in response to certain damage signals from the plants, and based on the construction using these Vir genes, you can actually get that transgene to go into the genome of the plant desired, as long as you have the right promoters set up. But we'll skip that one for now, but just showing you the basics of it. And of course, it results in glowy. So now we get on to the real meat of, the mammalian side of things. We're starting to get into the mammalian constructs. So remember, what was the big components of the bacterial stuff? We have a promoter, we have a transgene, we have selection markers, and we have origins of replication. All those are still present here. So it can be both grown in a bacteria, but instead of being coded to express in a bacteria, such as the previous one, we're having this express in a mammalian cell. So we need a different promoter. So, like I said before, same materials. We have an origin of replication. We have a selection marker. That way, when we are growing this up in a bacteria, we can take it out, we can isolate it, no issue whatsoever. It's self-selecting. If it doesn't have the selection marker, it dies from the antibiotic. Now, when it comes to mammalian stuff, we have particular mammalian promoters. In this case, this is a CMV. It's a strong promoter. And we have a transgene right after that. Now, the result of this is something we've done in our lab. I did this picture because you can see some that aren't totally transfected, but these are human cells. So I don't know if y'all know much about human cells. What you think might be a little bit strange about these human cells?
So they rim. Red.
Yeah, they're green. Most people aren't glowing green under UV light. So even at most raves, this is a great party trick. You essentially see that like the mammalian construct, we've transfected these into the cells. We'll go over that a little bit later to the slide because there's some problems. And a lot of this, towards the middle of this slide, there's going to be problems. And I think this is a lovely community of coders, biohackers, scientists, and AI enthusiasts. And there's problems that I'm putting out to y'all to solve. I'm just getting y'all familiar with this kind of work. So here's the thing. That being said, you can also do this in different cell types. This is from a colleague of mine. These are canine sperm cells. It's called sperm-mediated gene transfer. I'm sorry, guys, we're the easiest side of the genetic equation to modify. You can essentially use mammalian constructs, and you can essentially infect. This is how they do a lot of glowing mice, glowing cats. It's very easy to do because the sperms essentially are very easily to take up the genetic material using mammalian constructs, yet again. So RNA kind of things, essentially single-stranded nucleic acids. This is the same kind of thing that you see in the COVID vaccines. They're really popular, particularly for selection markers. They're made from DNA, so you take a plasmid, you essentially go through this standardized process, and then you typically enzymatically, and then you isolate them typically on a gel well. So these, this is an example I give. Typically, they're in two varieties. In this case, you have a gene of interest. That's what that stands for. And then you have essentially the RNA code and a poly-A signal tail at the end. Just big words, it's okay. It's basically you have the same thing that would have been on the mammalian construct, except it's been isolated after it's been coded out. So in this case, the A one is not what's called a non-amplifying one. So let's say if you want to have a larger effect, you can actually add in some additional bits to have the RNA make itself more upon delivery. So that way you have a stronger response. So keep in mind, these things don't last as long as some other constructs. For example, the mammalian constructs we just saw, those last, a couple days to, if you're lucky, depending on how you set it up, about a week if you're lucky, but mostly just a couple days. These are extremely transitory. You need to use a cold chain. They only last maybe a day or two post-transfection. So keep that in mind when you're using these things and determining what you want to do for your project. How long do I want this gene to actually express for? How long do I want it to last? Do I need a cold chain? These are all very important questions. How big of a response do I need? So this is just an example of some CHO cells that have been transfected with RNA. Essentially, it's similar to the HEK cells shown before. The blue is actually just a dye of the nucleus, but essentially they're just expressing GFP via transfection, and I believe these were polymer transfections. Okay, so now we actually get to the controversial bit, because who's seen Resident Evil or something like that?
Yes.
Just a couple people. So when we're talking about viruses, it's actually kind of limited, and this is one challenge I propose to any bioengineer going into this. Whether you want to look into this as something as more of a career bit, or just if you can actually put AI to the focus of this. We're only limited to 4.7 kilobase pairs on what you're actually able to insert with AAV standardly. So basically it's about a 3 plasmid system, or a 2 plasmid system and a 1 linear bit. So you essentially take these genetic bits, mix them up in some human cells, transfect them, and inside the cell, these different components are able to reassemble themselves into viral particles, which you can then isolate and then use either for therapeutics or for the production of other proteins, depending on how you've set up the system. So just as an example, typically you have a linear segment of AAV viral genome. Normally you have replicating proteins and essentially structural proteins. What we're going to do is we're going to remove those center bits on the linear strand and actually put our promoter, what we want to, the actual thing for a mammalian construct, and our gene of interest. On a separate plasmid, we'll have our promoters with the replicating bits and our actual structural bits, as well as a secondary plasmid with just some helpful things. These proteins essentially help assemble the viruses and get them ready to be produced. And we put all of that in a lovely human cell and let it transfect and grow up the virus for us. And then, kind of more boringly, what we mostly work on down in Honduras for our gene therapies, mini circles and mini intronic plasmids, essentially is a mammalian construct, but we remove or hide in via introns the essentially bacterial components of the DNA, whether that's the origin of replication as well as the selection marker. And essentially what you're doing there is essentially you're trying to make those already somewhat short-acting, what are they called, plasmids, essentially last longer. So you can get a couple weeks to months expression in vivo on some constructs to even what we have now with our phase trial up to a year and a half. Okay, so this is the meme of the hour, CRISPR-Cas9. This is the one that's probably most ubiquitous of being able to take things out, chop in DNA, homologous repair. And I think this is the one probably with the most development need, to be frank with you, in terms of its ubiquity of application as well as how it needs to be performed. Currently, this is the typical bit of CRISPR. Let's say you have our regular bit of DNA, you have a guide RNA, you have a target with a guide on that, with the corresponding sequence on the actual bit of, let's say, chromosomal DNA or even plasmid. It can be any variety of nucleic sequence that you want to, but as long as these sequences match up between the guide and the actual sequence, you can essentially target it for splicing, cutting out. Now, this can be used in two different ways. You can kill a gene, you can cut it out. A lot of times people go for the Kozak sequence or the start sequence. Without that start sequence, it's like trying to start a car without no key. So essentially, in this case, we have our sequence. It's the start of a gene. Let's say this is for some kind of awful protein that's making you sick. We have that guide essentially built up so that it will correspond to that awful sequence, and we'll make a cut there, and then the body by itself will essentially relink up those bits, hopefully. And secondarily, we can add an additional linear bit of DNA with overlapping bits that correspond to an area that we want to be repaired from it, with a new gene to be donated after the cut has been made. So, short of story is, these two bits, these complementary ends, end up with a little bit of ligation, end up repairing the bit of the DNA. So this is a standardized construct. Yet again, we see a lot of the same ubiquity. So we have promoters to express the actual Cas9 protein, followed by this is just a slippage sequence. So it will again produce a GFP bit for a tag. And then we have another sequence which essentially adds a promoter for the RNA sequence. So we have a standard little loop bin for the non-coding bit of the guide, and then we actually have the corresponding guide that will link up with whatever gene we're trying to target it. Let's say, now this can be used in almost any setting. You can use this on plant cells. You can use this on human cells. You can use this on bacterial cells. I had a project I was working on where they wanted to insert a particular protein into the genome of a lactobacillus species. And so we chose CRISPR with homologous repair to essentially build up a little scaffolding that we wanted inside that genome. You can also cut out things. For example, there was another project I was helping with where they have spirulina, where they want to cut out some particularly bitter tasting compound producing proteins actually in the genome of the organism. So you're able to go into the genome, cut out what you don't want, or add a particular thing that you do want, and then get out. And of course, the longer the guide RNA, the more accurate you can be. And of course, more and more generations of these things are becoming more and more accurate, but a lot of times it still isn't fully accurate. And so If anyone has any bit, I'm sure the next speaker with bioinformatics might be able to help, or some of y'all AI folks. These are all prime targets that need a little bit help on.
Actually, at my workshop, we used another insertion with CRISPR prime editing, not tools that you mentioned, but the certain method.
Yeah, there's a lot more than what I'm going over in just this talk. It's a very wide field, but they're generalized. This is essentially one of the more basic variants of it. So now we're going, how do we actually get all these different constructs in? Maybe this would probably have been good. There's a couple different manners. You can look at these either chemical insertions, physical insertions, and all these are different methodologies, each working for a different type of cell type. So, for example, heat shock, mostly this is just for E. coli, bacteria. You're essentially using a little bit of salt to kind of stop up, calcium salt to stop up the cell, and then with heat, actually like expand it, crack it open a little bit, and you're allowed to get plasmids into the cell, mostly for, let's say, if you want to produce more plasmid or a particular protein from the E. coli. There's magnetofection. So you can use this on mammalian cells. You can also use this, I believe they've actually got this working for certain whole body apparatuses for mice. But essentially you're using a very strong magnetic pulse to mess up the charge differential on the outside of the thing, kind of crack open the cell membrane, allowing virus or RNA particles or plasmid constructs to be inserted into it. And of course, electroporation. Similar manner, except you're using this more on, you can use this on harder cell types such as plants, certain types of bacteria. You can also use a weaker shock, zap zap. I always like to think of myself as Dr. Frankenstein every time I do it, when I pull the little switch on the little instrument. Just because it is essentially, you're doing an electrical pulse, and that is essentially making little micro tears that you can get plasmid or RNA into the actual cell based on those tears. Now this is another area of interest because it's not fully, on certain cell types, everything's nice and figured out, but there's no real continuum to be like, if I have a new cell, it's almost trial and error whether I kill my cells or not. Every time you come across a new cell line or particularly a new species of cell. And of course, everyone's favorite lipid nanoparticle. These are little tiny lipid-like balls of, that essentially you can have them fuse into the membrane and essentially get them to deliver either a drug, RNA, DNA, these kinds of things. This is the same stuff that was used in the COVID vaccines. A lot of times this is used in the production of AAV viruses with lipofectamine. And of course, the one that we're using, sometimes these can be a little bit cytotoxic, but these are cationic polymers. Think PEI, certain types of PLL. These are just chemical polymers that essentially you can act as like a tumbleweed bouncing through a cell where it electrostatically is bound to the cell essentially eats it, and then as it's being chewed up, it can deposit the DNA or RNA into the, the insides of the cell. So, okay, who's still with me? Clap if you're still here. I know that was a lot. But that was just to give kind of a brief introduction. That way, after you leave here, you can look into things that particularly interest you, as well as trying to apply AI, different types of technologies that you're more familiar with to these various areas of application. I'm going to give y'all some resources that are free, 90 free on some of them, such as Benchling. This is you can just sign up now. You can go in, you can play with different constructs. You can actually create any, you can make entire genome edits or anything like that, or like plans. You can have it make viral vectors. It's essentially an open source kind of DNA compiler, if you will. It's where you can actually go in and code what you want to code, or import other DNA files from colleagues or other online resources. One of them I really highly suggest is AdGene. We are all but students in comparison to the masters. The benefit about AdGene, you can actually order these both. It depends on whether if you're a nonprofit, you get the cheaper price, or a university, or you can pay a higher price a lot of times if you're actually for-profit. But the big thing is you get access, you can actually take a look at the publication, the construct it comes from, and you can use the information gleaned from this construct into whatever you're working with. There's hundreds of different types of plasmids, constructs, everything from AAV virus, lentivirus, plasmids, agrobacterium constructs, mammalian constructs, and it's all, the whole sequence is right there for the taking. And you can go in, take that to something like Benchlane, mess around with it, and actually produce something of novel value using all these different resources by putting everything together. Another one that they showed in the the CRISPR workshop earlier that you just kind of want to get into it. Like again, gene of interest, promoter, same kind of thing, but essentially kind of hold your hand as you walk through in just basically setting up a construct for the first time. So it's a very handy resource, especially as you're just getting into doing it, and you can even order the constructs from Vector Builder straight to, depending on where you are. As well as once you've gotten more accustomed to it, you can go for full total synthesis on different constructs, different types of genes. You can order fragments from Twist Bioscience. It's 8 cents a base pair, which is fairly cheap in terms of industry standards.
No. Or no? Oh, clearly yes. Hi, so this is meant to be a session called Bioinformatics, which is really vague, I must say. But I'll be actually talking about something quite a bit more specific. So I'm Lucia Klaric, and I'm a statistical scientist at Humanity. And today I will walk you through how we are building and how we built a personalized recommendation system to slow down aging. So my parents are roughly of similar age, 67, I think. And my dad, on a good day, can walk 2 kilometers without losing breath. My mom, on at least a few times a year, runs ultramarathon. So they're quite a similar age chronologically, but clearly they are biologically of different age. And scientific community might not have a consensus of an exact definition of the biological age, but we sort of all intuitively know that it exists and that it sort of should be measured. And indeed, there were various efforts in the community trying to build so-called omics clocks of aging. And we can see here a small graphic that sort of shows in different colors different omics data sets that were used to predict chronological age, but then, more importantly, to see how their predicted age, called biological age, actually associates with different health-related phenotypes. So, for example, there's a number of aging clocks, omics aging clocks, that are associated with cognitive decline, coronary artery disease, blood pressure, diabetes, physical dysfunction, mortality, BMI, progeria, and all this various different, not all of them are even listed here. And so this is very important research because we'd like to know who is at higher risk of developing a disease sooner or dying sooner. And by knowing that and using different omics technologies to quantify that, we are also sort of measuring different aspects of aging biology so we can potentially develop therapies for that. But there is a more important question here. Well, not more important questions, but as I've been aging and observing the people around me, I realized there must certainly be something that we can do now. So it's really good to have clocks, but actually what can we do now to slow down our aging? And I'm not talking about things that we heard a few minutes ago. I'm talking about more of the daily, everyday things that we can do. I'm sure all of you here would have various suggestions. Looking at my parents, I would say drink less, smoke less, and run more. And it's actually something along the lines what doctors have been telling us and research have been showing us for a long time. We are all very well that physical activity associates with biological age. I really like this paper because it had a title, How Little is Enough? And it's quite interesting because it was done on 72,000 participants from this publicly funded study called UK Biobank. They gave watch accelerometers to 72,000 people to wear them for 7 days. And through this data, they sort of assessed their frequency of average vigorous physical activity levels in a week. Then they went through their electronic health records with a 6-year follow-up and just looked sort of, did people contract diseases or unfortunately died since they wore the device? And can we tell the difference between the people that were more physically active and those that were less physically active? And indeed, we can see here an example of 15 years of risk, 5-year risk of cardiovascular disease incidence. So this is a group that had done no vigorous physical activity. And we see, and these are the groups that done various levels of physical activity weekly, and we can see there's a difference in the risk of contracting cardiovascular disease. And sort of the take-home message from this paper was 15 to 20 minutes weekly of vigorous physical activity were associated with 18 to 24% lower all-cause mortality with an optimal dose of 50 to 57 minutes a week. Quite precise, I would say 57 minutes, but anyways, it gives a rough idea what to do. There's another, a bit older story from 10 years ago, 11, 12 years ago, published in Lancet, where they looked at roughly 500,000 people from Taiwan. And it was a questionnaire-based data where they separated participants into 5 categories based on exercise volumes. They had people that were inactive, low, medium, high, or very highly active. And then did a similar thing, looked at the 8 years of follow-up and looking at all-cause mortality. And we can see here again that sort of we have compared to people that were inactive. This is on the y-axis, you can see all-cause mortality reduction. Meaning, and if you look at it, so this is 50% and this is 0. So we see that people that did vigorous daily physical activity, for example, 50 minutes, they had almost 45% reduction in all-cause mortality. If you look here, even moderate physical activity is important, and this is sort of an average. So basically, they take away home message was 50 minutes of daily vigorous intensity in physical activity resulted in a similar cause mortality risk reduction, 25% as 60 minutes of daily physical activity at moderate intensity. So now we can also sort of like start waging how much of what can I do that suits me more. Let's turn further. What doctors always tell us after they stop telling us smoking is fine in 20s, run more and eat less. There's also a vast research on how nutritional habits affect aging. So control meal timing expands health span. So if you look at this paper from New England Journal of Medicine, the main message is that intermittent fasting has broad spectrum benefits for many health conditions, such as obesity, diabetes, cardiovascular diseases, cancers, and neurological disorders, all abroad, all the spectrums. There's another paper that said maintaining a regular daily feeding fasting pattern may be sufficient to optimize an individual's physiology and decrease their risk of disease to extend health span. And we can even go further on, and there's research showing that metabolic switch is flipped following 12 hours of fasting. So we can sort of, from the scientific literature, start learning what's important and good for us. So we've seen from the research, and we know from our own lives, that lifestyle factors predict mortality, but knowledge of risk factors alone isn't enough to motivate people towards healthy behavior. So presenting risk information on its own, even when highly personalized, does not produce strong effects on health-related behaviors or changes which are sustained. So there is a need to effectively moderate our behavior. And luckily, humans have a sort of a characteristic that their behavior can be supercharged through gamification. It turns out that we as humans, like when we set some goals, we can monitor those goals, we get a feedback on performance, we get cues what to do, and we can plan what to do. And it turns out from research that in such cases when we can actually gamify what we need to do, we are more likely to actually stick to the plan. And this is exactly what humanity has been doing. So we have created the humanity score that drives behavior change through gamification. And sort of this is almost a spoiler alert. The humanity score, which you can see example right here, is bootloaded with robust peer-reviewed research, as some of those that I just showed, research settings, and then stratified dynamically as more user actions and results are being monitored. So we started from what the research tells. We collected loads of data from our users, and now we started making personalized recommendations based on what we learned from our users, what works best for their biological aging. It is calculated as composite score from movement, mind, recovery, and nutrition. So sort of we separated these actions in 4 subsets. And what's important, it rewards and promotes positive health behavior. You see here, like, what are they? And you get like prompts, keep on, and so on. So this, provides an engine for users to track and reward their health behaviors. So how do we actually build it? It's built from 4 independent scores. First one is movement. So research has shown that physical activity is well linked to reductions in disease risk and mortality. And so what do we actually do? We passively monitor heart rate if you have a wearable, and step rate if you have only phone, as well as any manual. You can also manually record any activities that you did that may not have been captured by any of your devices. And this is all scored as part of the movement. Then we have a recovery. We all know how important it is to sleep well. So sleep and recovery techniques are essential to maintenance of physiological function. I also heard that Australia is supposedly thinking of starting to test drivers whether they are sleep deprived. Supposedly being very sleep deprived is equivalent of being slightly drunk. So I think it's still quite controversial, but it's sort of, they're saying if you sleep for less than 5 hours, it's equivalent of drinking more than 2 beers. So we should sort of be careful who we let drive. So sort of sleep is very important. And so how we record this? Optimal sleep duration and actions are known to improve quality and improve recovery score. So we track how much you sleep, either manually or through an app, and which other activities you do that can count as recovery, and we reward you for good behavior. Nutrition, intermittent fasting, and balanced diet is associated with longevity. So we track overnight fasting duration, and we also track how much snacks you eat. And fewer snacks, better your nutrition score, and longer fast, likewise. And the last part is related with mind and health. And if you were there at the forum, there was sort of a mention that we should start talking a bit more about the mental health. So in the mind part, we record your meditation and mindfulness, as well as actions known to improve mental well-being, to improve your mind score. So overall, the humanity score, we calculate all these 4 things together, and you get a daily score that ranges from 0 to 100. And the app and design and experience is built to reward healthy actions and create dopamine feedback loops. So sort of it wants you to do good because you get this dopamine feedback loop because you did so well. And this sticky user experience supports users to healthy behavior. So currently, in past, the humanity score was bootloaded from robust peer-reviewed research. But as I said, can we now, when we have a big user database, use the data we gather from our large user base to tune the humanity score to be personalized and more strongly rated with biological age? Spoiler alert, the answer is yes. So how did we did this? This is just a brief overview. I'll go through the details of every step. We first calculated BAA, biological age acceleration, which is simply just chronological age minus your biological age. We extract your data that you recorded, or we extracted from your phones and devices automatically, and then we simply calculate the relationship between BAA and your actions, and in that we sort of can then manipulate how much of each action you should do that would be more strongly associated with your biological age. So first, how do we calculate biological age? In humanity, basically we have currently four different sources of biological age. First one is based on the patterns of movement and heart rate, which is in collaboration with a company called the Giro. So this uses your step rate, heart rate, and applies complex machine learning methods to calculate your BAA. We have clinical blood biomarkers. These are the things that you can go to your doctor and ask to be measured, like cholesterol, triglycerides, red blood cells. There's a whole list of them. So we developed in-house machine learning model based on the UKBab and data that I already mentioned before. We have some DNA methylation data, biological age based from methylation data from the chronomics, and we have our own humanity score based on beneficial actions of our users. So there are these four sources, and basically we combine them in one average what's your average biological age. Though in theory, we could also focus on each one of these separately. So that's step 1. Step 2 is extracting the data from, looking at the data that we obtain from our users. And so as I said, first we calculate movement, and this is captured by automatic extraction from either phone or a wearable device. You see here how it looks like in the app. And we have 2 types of movement data. We have just plain steps, and we also have training intensities, and they are based either on step or heart rate. And these are separated, as similar as in those papers that I showed, either in low intensity training, moderate intensity training, and high intensity training. If we look at the data itself, on average, our users daily make 5,200 steps. And here we see the distribution of different training intensities. First thing to notice is to pay attention to the x-axis, and I don't know if you can see far away, but this is 1.5 hour, and this is 6 hours, and this is 4 hours. So we can tell that when training, our users mostly do low intensity training. So this could also be biased because some people actually have to walk a lot to work. When we look at the moderate intensity training, on average our users do 22 minutes, but some do up to 3 or 4 more hours. And average intensity training, duration of high intensity training is 12 minutes with a maximum of even longer than 1 hour. So that's movement. The next one that we calculate together is nutrition. And our users are prompted to daily record length of overnight fasting, number of snacks eaten in a day, and number of meals eaten in a day. And if we look at the data itself, we can tell that majority of our users on average fast for up to 13 hours. So we can tell that our users mostly skip breakfast, but there are also users that fast for longer than that, and there are also occasionally people that fast for a full day. Of course, here the hours of fasting maximum is 24 because one day you can only have 24 hours. If you look at the number of snacks, majority of our users have 2 or fewer snacks a day. But there are some that really should think about having 9 snacks a day. That's a good lifestyle, perhaps, but no, not maybe the healthiest one. And on the other hand, a majority of users, as we probably might expect, have 3 meals a day. As far as the recovery is concerned, we prompt users to daily record numbers of hours they slept, or it can also be automatic, extracted from the app that you connected with our app. And we see here that, so this is 7 hours, but on average the users sleep a bit longer than 7 hours, which is, I would say probably good, but they do say that optimal sleeping between 7 and 8 hours. Here we also record time spent exposed to natural light, so just not being in a room like this, but rather being at the amphitheater and in the open space. And also, these are basically binary actions, time in hot or cold temperatures. So if you had a hot plunge this morning or at 1 p.m., you would record it as action that you did today. In the mind category, we record mindful minutes, which can also be either manually recorded or from the automatic extracted from the apps. We also ask people how they feel like, and we don't score feeling good more than we score feeling bad. We just think it's really good to reflect on your day, and to record how you felt. It might help your overall mental health. And here we also have time spent in nature, so just another binary action, just have you been outside for at least 15 minutes. And if we look at some distribution of the data from our users are on average spend 15 minutes meditating or doing mindful actions. And I was quite pleased to see this one because it looks like our users are actually quite happy. So on average, they score 68, and there's a whole bunch of users that feels amazing every day, which is nice to hear. So there's the last step now, calculating. So we calculate the BAA, we look at our user data, and now is the last step, how do we actually calculate the relationship between BAA and actions? Well, it's actually rather simple. We look on one axis our users' behavior. Are they more active or are they more couch potatoes? On the other axis, we look at their biological age. Are they looking younger based on those 4 ages that we looked at, or are they looking a bit older? And sort of we just plot one against each other, and then we try just to simply, in the most simple way, put a line between. And this will tell us, slope of this line will, and spread the points around the line will tell us how important movement is for biological age acceleration. This is just a simplified explanation. And then based on that, we can try sort of rewarding you more for the things that are more important for your biological age. And so we model, and so we do this in a few things. First, what we do, we model your action behavior versus your future BAA. So how you did this month compared to BAA in 5 weeks' time. So what we do now affects what happens with your BAA in the future. We do it by accounting for relationship between actions, meaning that when we have BAA, we are taking all the actions into account at the same time. And we also incorporate prediction of how changes in your action affect changes in your BAA. And so this is what we learned. So previously, our age score was based on solely on literature-based research, and this is how our tuned model would look like. And so we here see the effect on age score. We see that high-intensity training, if you look at overall our user base, is one of the most important things you can do. Low-intensity training is the next. Snacks are really bad. We don't recommend almost any snacks. I guess there could be exception if it's fruits, though this will be recorded in the app in the future. It's not at a minute. Steps are very important, and sleep is very important. And then all these other actions contribute with varying levels. So this is good. There is good data, there is a good research, we have improved our scores, but you are not the general populations. Can we do better than that? Can we give recommendations for you personally? And to do that, we did the following. We stratified our users based on their physical characteristics, and this is a current beta version. So age first, are you older or younger than 34? 34 because that's just the median age of our users. Are you female or male, self-declared? And your weight through BMI. So are you, is your BMI lower than 25 or higher than 25, which is by World Health Organization classified as overweight. So we group people in our data together based on their characteristics. So for example, we here take men of BMI lower than 25 that are younger than 34, and we repeat what I did previous. We look how the BAA in this group of people is related with the actions they record. And then we did the same for men who are higher BMI and younger than 34, so on and so forth. And there's in total 8 different groups based on these 3 characteristics. And this is what we gain. These are our personalized scores. So how to look at these graphs, because there are a lot. They're ordered based on what's important for female older BMI. So the quickest way of looking at this graph is just if the columns are in the right order. So here we see high, slow, slow. So here we see a change. So that would mean that sleep is more important than intensity low for female older with higher BMI. And this gave us a really nice sort of quick way of visualizing how personalized this score is. For example, if you look at a female younger than 34 with BMI lower than 25, actually they should do more intensity low training than intensity high training, and really they shouldn't, the snacks are really bad for them. In the same way, if you look, for example, for male that are older than 34 and have BMI less than 25, if you look at this, intensity high is more important than it is, for example, for a female. And perhaps the easier way to see this, if we just take one of the groups as a reference. So I just took, what if we take this as a reference, how different now everyone is for female, older, BMI less than 25. And if you look at myself, I should be, I should be fasting more, I should be having fewer snacks, and I should sleep more, which I think with the fasting and snacks I could totally agree with. I'm a bit more of my father than my mother. And if you, for example, compare with a male older BMI, you can see that intensity high is more important for them than it is for a female. And sort of with this, now we can personalize and say, depending on which of these categories you belong to, you are sort of, these are your recommendations.
And you found it.
And just the final comparison that I'll show, this is how personalized this is, because I first show you the model that contained all the user data. And this, we are now looking at the differences of that model for all the user data between each individual category. And we see it's sort of that there are quite striking differences. If we just used the model that was based on all population, we would see that, we would never see that, for example, that for female younger, it's actually more important to do more intensity high and less intensity low compared to what they would be suggested if everyone was treated the same.
But they had
And so with this, I would like to conclude that lifestyle interventions that we capture in humanity are associated with biological age. And we can use the data that we obtain from the users to create personalized recommendations for our users. So sort of, depending on which category you're in, you would benefit more from doing one than the other. Of course, if you're doing well, you are likely to change between the categories, at least with regard to weight. So sort of there's always a scope. Your recommendations keep changing as well as you age and so on and so forth. So this is only sort of a first step. There's so much more that we could do. This stratification is quite crude. We all know that there are, as I already said, my mother is way more fit than potentially an average person of her age. So in future, it would be really good if we could create these strata, not necessarily based on visible characteristics, but we can use some unsupervised methods that could define users that look most similar together and based then modeling on their behavior. Another thing that we noticed through this work, that the strongest associations so far are from movement and nutrition, so you benefit most from those, while other actions are also important. We currently just need more data to more fine-tune how much of these other actions we would really need. And because of the interpretability of the model, we are currently only using linear relationship. There's so much more that we could do. If we stepped further, we know that the life is not linear, but this is sort of a good start. And also, if you think about it as a system in general, currently there are 4 biological ages, but there's so much you could do. We could incorporate any other biological age out there into our models. And then you could potentially even choose, do you want it modeled for your composite age, summary of all your age, or you want to focus, I prefer to focus on metabolic age rather than, glycan age and so forth, so forth. So I hope it is I convinced us this is we can use the data from the users to learn how to do better for ourselves and to keep being younger. Thank you. I'm happy to take any questions.
yes.